So, hello students, welcome back to Vedantu NEET English. This is Basavaraj Sir, your biology maths teacher. Students, today is going to be a very special class because we will be doing something called as monocot versus dicot. Now students, I know in your syllabus there is something called as root, stem, leaf. Yes, monocot and dicot. And this has always been a very difficult way to learn that monocot and dicot, what is the difference? And in examination, which type of question you can expect from monocot and dicot? All that confusion will be clear from today's session. And students, let me tell you, all of you are my students, you already know how we approach anatomy, right? We approach anatomy with the help of diagrams. We see the diagrams and we understand from diagrams and write what is seen in the diagrams and nothing more, nothing less. Okay. So with that being said, students, quickly let me know in the chat right now if I'm audible and visible to each and every one of you. Quickly let me know in the chat, all of you, if I'm audible and visible. Yes. Good evening, good evening, good evening, every one of you. Good evening. 1 second. Let me just down. Okay. Perfect. 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 Okay. Now, students, I'll ask you a simple question. All of you. Do you see this particular sectioning on the board? Can anyone tell me which sectioning is it? Is it your monocot or dicot? Okay. Quickly. This is a monocot section. How do I know that? Because can you see scattered vascular bundles, water cavities, and also towards the corner we have small and many vascular bundles. As we come towards the center, large and bigger vascular bundles. Don't worry. If you do not know, I will teach you what exactly is that in today's lecture. Now students, before we start to the sectioning, before we go to sectioning entirely, does anyone know what is the basic difference between monocot and dicot? The basic difference between monocot and dicot. Tell me, anyone knows? The basic difference between monocot versus dicot. Monocot versus dicot. What is the major difference? The starting difference. It is none other than the number of cotyledons. Yes or no? It is the number of cotyledons. Number of cotyledons. Now students, all of you are aware that the number of cotyledons vary both in the case of monocot as well as dicot. It varies both in the case of monocot and dicot. Especially students, if you are aware that in the case of monocot, there is a single cotyledon. How many of you know that? In the case of monocot, there is a single cotyledon called as scutellum. Yes or no? Scutellum is a single cotyledon. But in the case of dicot, there is two cotyledons. There is two cotyledons in the case of dicot. Two cotyledons are there in the case of dicot. Now, but in examination, students, sometimes what happens is they will ask you the question. They'll ask you the question based on the example given, right? So you need to know a few basic examples of monocot and dicot. Let me tell you, based on which question can come in your examination. Okay, understand. For your monocots, one of the famous examples is the grasses. All of you are aware, the wheat, barley, rice, right? The wheat, rice, barley, maize, all of these are called as your grasses. Wheat, rice, barley, maize are called as your grasses. Apart from that, one more example which they can ask you is lilies, or they can also give you the examples of your orchids. Sometimes they can also give you banana and onion. Yes, banana and onion are also a type of monocot. They're also a type of monocot. But when you come to dicot, they'll always give you the example of roses, rosaceae members, right? Roses, or it could be legumes, leguminaceae, sunflower, helianthus, that is your astraceae, sunflower. So all of you need to know the basic difference between monocot and dicot, and also few examples based on which the question can be asked. Yes or no? Now there is a lot of difference between monocot and dicot. Stem, root, leaf, even the pollen development. There are so many differences. But what we'll be learning in today's class is the basic difference between stem, root and leaf. Can we start, students? Can we start? the basic difference between the stem, the root and the leaf. So with that being said, let's get with the first diagram on the board right here. Now students, the first thing which we are approaching is actually the dicot root right here. Okay, dicot root. Now, can anyone tell me which is the outermost layer found in the dicot root? Quickly tell me, students. Outermost layer. See, do not by heart any of it. Just see and tell me which is the outermost layer here from the entire section. You can tell me, sir, the outermost layer which is present here in the case of dicot root is the epidermis. Yes or no? Tell me, students. The outermost layer is the epidermis right here. It is the epidermis. Okay. Outermost layer is called as the epidermis right here. Okay. It is epidermis. Now, obviously, it is the outermost layer. Yes, sir. It is the outermost layer. Outermost layer. Outermost layer is called as your epidermis. Now, tell me, students, is it a single layer or is it multi-layer? Tell me. Look at the diagram and tell me, is it a single layer structure or is it made up of many layers? See this diagram right here. You will tell me, sir, it is made up of a single layer. Okay, it is made up of a single layer. Single layer structure. Now, students, you need to understand the epidermis is also called as, it is also known as epiblema. It is also known as epiblema. Epiblema. This is the other name of your epidermis. So I'll write here also. Epiblema in the case of root. Epiblema in the case of root. Okay. Now, students, you tell me, in the case of the root, do you need cuticle? Do you need cuticle by any chance? It is a root. It is under the soil. So obviously, you do not need a sunscreen. If it is already under the... Tell me, students, if you are sitting inside your house, will you apply sunscreen? Obviously not. You only need sunscreen when you are outside the house. Okay. So technically speaking, the roots does not have cuticle. Can I tell? The cuticle is absent here. Cuticle is absent. Cuticle is said to be completely absent here. Okay. Cuticle is said to be absent here. Cuticle is absent. Now, what about stomata? Obviously, it is not exposed to the sun. There is no gases exchange here. So, stomata also is completely absent here. Stomata is also absent. Stomata is also absent here. Stomata also is said to be absent. Yes or no? Now, students, look at the diagram. Look at the diagram and tell me, do you find anywhere any form of intercellular spaces anywhere? Tell me, all of you. Anywhere, do you see any form of intercellular spaces anywhere? Obviously not. So can I tell the intercellular spaces, ICS, intercellular spaces is completely absent. Okay. So apart from all that, the one thing which I have not written here, which is already seen in this epiblema or epidermis, is can you see any appendages coming out? Can you see here some form of appendages coming out? Yes or no? Tell me. Yes or no? Can you see? Yes, sir. I can see it is outermost layer, single layer. It is also known as epiblema. It is also known as epiblema. Also known as your epiblema. And sir, it is cuticle is absent. Stomata is absent. Intercellular space is absent. But apart from all of these points, the one point which is missing here is the unicellular root hair. It has something called as unicellular root hair. Unicellular root hair. Unicellular root hair. Done. Now, these are the following points which you need to remember for your epidermis. Done. Tell me, students, do you need to remember it? Yes. All the following points are for your dicot root, the epidermis. Done, all of you? If it is done, thumbs up in the chat, all of you, quickly. If there is no doubt till here, thumbs up in the chat quickly. Okay. So unicellular root hair, intercellular space is absent, cuticle is absent, stomata is absent. It is also known as epiblema, single layer, outermost layer, epidermis. Okay. Now, students, you need to remember this root hair is also called as your trichoblast. That is not given in your NCERT. Trichoblast is not given in your NCERT. Okay. Now, till here, clear. Now, students, which is the next layer? Now, there's a famous question here. The famous question is, do you find hypodermis here? Do you find hypodermis here? No, sir. Hypodermis is completely absent. It is completely absent in the case of your root. Root, hypodermis is absent. Hypodermis is absent here. Okay. So if hypodermis is absent, which is the next layer? Can anyone tell me? All of you see this particular diagram and tell me which is the next layer. You will tell me, sir. The next layer is something called as your cortex here. Yes. So next layer is called as what? Cortex. Cortex is the next layer. Cortex is the next layer. Now students, look at the cortex and tell me, do you think the cortex is a single layer or multi-layer? See, 1, 2, 3, 4, 5, 6. Can I tell it is multi-layer? Yes, sir, it is multi-layer. It is multi-layer. It is said to be multi-layer or many layers. Or many layers. Okay. Now, if it is multi-layer, it is also made up of thin-walled parenchyma. Thin-walled parenchyma. Thin-walled parenchyma cells. Parenchyma cells. Thin-walled parenchyma cells. Now, students, in this entire diagram, do you see a small gaps or holes? Do you see a small gaps or holes there anywhere? Can you see, students? Do you see any small gaps? Can you see small it is present? Somewhere it is present. Can I say somewhere it is present here? So can I tell intercellular space is present or absent in many cases? Or absent? Or it is said to be very less. Very less intercellular spaces, and that is what cortex is over. Cortex is over, that's all. Cortex is over. Okay. So what are the points for cortex? Multi-layer or many-layer, thin-walled parenchyma, intercellular space is present or absent in many cases, but even if it is present, it is very less intercellular space is present here. Done. So epidermis, all the following points. Cortex is the points which you need to write down. Okay. Now students, the innermost layer of cortex. The innermost layer of cortex. Can you tell me? Look at the diagram and tell me which is called as the innermost layer of the cortex. The next layer, the pink color one. Can you see? The pink color one. Tell me, students, which is the next layer? The innermost layer of cortex is called as your endodermis. Yes or no? So then we have the endodermis. Then we have the endodermis. Endodermis is the innermost layer. So first of all, you should understand here is it is the innermost layer. It is the innermost layer of cortex. Of the cortex. Innermost layer of the cortex, that is your endodermis. That is your endodermis as the innermost cortex. Now students, tell me, is it single layer or many layer? Look at the diagram and tell me, students. Look at the diagram and tell me, is it single layer or many layers? Is it single layer or is it many layers? Tell me, students. This one, this line, pink color one. You will tell me, sir, it is single layer here. So write down. Write down. Single layer made up of what? Made up of parenchyma. Made up of parenchyma. Single layer made up of parenchyma. That is your endodermis. Endodermis, single layer. Now students, in this single layer of endodermis, do you see any form of small gaps or holes? Do you find any type of intercellular space here in the endodermis? Look at the diagram and tell me, students. Look at diagram and tell me. Do you find anywhere? Yes or no? Tell me, students. Obviously not. So intercellular space is completely absent. Intercellular space is completely absent here. Absent. Now students, there is a special shape of cell here. See, apart from these cells are much more round shape, or like curvature is there everywhere. Yes. So what we call here is the endodermis has a special type of shape, and the special type of shape is called as barrel shaped. Understand, students? If this is the cells, do you see? All the cells are in somewhat barrel shaped. This is called as barrel shaped cells. They are called as barrel shaped cells. So what do we have here? We have your barrel shaped cells. Barrel shaped cells. This is your barrel shaped cells. Now students, you need to understand one more small point here. The small point here is that in this barrel shaped cells, there is some form of depositions. For example, I'll show you. Let's take this bottle only. Okay. For example, this is a barrel shaped cell. For example, this is a barrel shaped cell. In this barrel shaped cell, there is some form of waxy material, also called as water impermeable substance, called as suberin is there. What is there? Suberin is there. For example, see this. This is called as your barrel shaped cell. Okay, barrel shaped cell. In this barrel shaped cell, there is suberin depositions. Now I want you to understand how is the suberin attached there. Listen to me very carefully. This is called as your tangential, radial. Okay. So for example, this is a barrel shaped cell. In this barrel shaped cell, tangentially there is suberin depositions. Radially also there is suberin depositions. So what is the students? Here we have something called as water impermeable. In the barrel shaped cell, we have something called as water impermeable. Water impermeable substance, waxy substance called as suberin depositions. They're called as what? Suberin depositions. Okay, suberin depositions. Now students, now suberin is formed in only one spot, or is it present as a complete layer? Can you see here? Suberin is not present randomly anywhere. It is present as an entire layer. Can you see? As an entire layer, suberin deposition is there. This entire layer of suberin deposition, or can I say it is present in the form of a strip. Can you see? It is present in the form of strip and strip. So suberin depositions in the form of a strip is called as Casparian strips. It is called as what? Casparian strips. Called as Casparian strips. Okay. So what did we learn here? This is your entire endodermis. So in endodermis we learned that here we have, it is the innermost layer of the cortex, present as a single layer, no intercellular spaces, barrel shaped cells. In the barrel shaped cells, here we have a waxy water impermeable substance called as suberin, and suberin is in the form of Casparian strips. Suberin is in the form of a Casparian strips. Clear all of you? Yes. Hello Philo, Sukanya, Madhumati, Sneha, all of you clear? In the form of a Casparian strips. Long Casparian strips is there. This one is the Casparian strips. Beautiful. Now students, imagine you are in your school. You are in your school or college anywhere. Can anyone randomly come inside, or do they have to go under a check? They have to undergo a checking. Also, I'll give you one more example. If you have to catch a flight, do they check before if you're the right person to enter? Yes. Similar function is with the plasma membrane also. Plasma membrane will not allow randomly anyone inside. It will do a checkpoint. Yes. So within the plant root also, in the root system also, the endodermis right here, this right here will act like a biological check post. It will act like what? It will act like a biological check post. Very extra point. So write down. It is a biological check post. Biological check post. It will make sure that all the proper water is coming, minerals are coming. So it will check. It will not randomly allow anything inside. It is the biological check post. That is your endodermis. So we learned about your epidermis, cortex, and the endodermis. Innermost layer, single layer made up of parenchyma. Intercellular space is completely absent. Barrel shaped cells are present here, and they have a suberin depositions in the form of Casparian strips. Now students, tell me, all of you tell me, which is present inner to the endodermis? Can anyone tell me inner to the endodermis what is present? Look at the diagram and tell me. Look at the diagram and tell me, students. Look at the diagram and tell me what is present inner to the endodermis. You can see one small cycle here. Can all of you see this small cycle here? That is called as pericycle. That is called as your pericycle. So pericycle is present inner to the endodermis. Inner to the endodermis. Now tell me, students, is the pericycle few layer or single layer? All of you see the diagram. All of you see the diagram and tell me this pericycle right here. This pericycle right here. This one right here. Can you see it is a few layer, not just a single layer? It is a few layer here. Pericycle. Yes, sir. It is a few layer. Pericycle is said to be few layer. It is few layer. Now, unlike your other parenchyma, your pericycle is actually thick-walled parenchyma. It is thick-walled parenchyma. Thick-walled parenchyma. Thick-walled parenchyma. That is your entire thing. Now, students, you need to understand the pericycle has two main roles. The pericycle right here has two main roles. Apart from these two roles, nothing else. Okay. The first role is called as initiation of lateral roots, and the second one is the growth of the cambium during the secondary growth. So write down. So this pericycle has two main functions. The pericycle has two main functions. The first one is called as initiation of lateral roots. Initiation of lateral roots. Lateral roots. How many of you remember lateral roots from the morphology we learn? Yes or no? And students, remember, they are coming from the pericycle. Roots are coming out of the pericycle. The roots are coming out of the pericycle like this. From pericycle, they are coming out. Lateral roots are coming out of the pericycle. So can I tell they are endogenous in origin? They are endogenous in origin. Endogenous in origin. Endogenous in origin. So that is the main function of your pericycle. Initiation of lateral roots are coming out. Lateral roots are coming out. Now the second function is that is the growth of vascular cambium. Growth of vascular cambium. Vascular cambium during secondary growth. During secondary growth in these cells. Okay, so that is the main function of pericycle. The main function of pericycle is that the first thing is it's a few layer thick-walled parenchyma. First main function is initiation of lateral roots, endogenous in origin, and the second thing is that growth of vascular cambium during the secondary growth in these cells. Secondary growth happens in these cells. That is your pericycle. That is your entire pericycle. Okay. Till now, any doubt, anyone, students? Quickly ask me. I hope you're writing down the notes. Write down these notes with me. Keep them with you, and it will benefit you in a very long run. Because when the exam is near, it is very difficult to read the entire NCERT. Rather, it is very easy to just read these points and remember. Okay. So I hope this is clear till now. Pericycle. Okay. Now, students, inner to the pericycle is the entire vascular bundles. Now, students, how many of you know the function of vascular bundles? Different types of vascular bundles. How many of you know different types of vascular bundle systems? Vascular tissue systems. All of you know. I'll give you a small recap. First of all, there is two main categories. First of all. Okay. In the first two categories, what is happening? Understand. Usually, all of you tend to forget this, so I'll give you a small recap here. Okay, See this. Now, there are two main categories here. One is called as your radial, and other one is called as your conjoint. It is called as conjoint. Understand, all of you. What exactly happens in the case of radial? Understand. In the case of radial arrangement, we have xylem, phloem, xylem, phloem, xylem, phloem, xylem, and phloem. Now, in the case of radial arrangement, do you see the each vascular bundle, xylem, phloem, are in an alternating manner? Yes. Are they in their own radii? See, xylem is present in one radii, phloem is present in one single radii. It is called as radial arrangement, and it is found in the case of roots. In the case of roots. Now, what is conjoint? What is happening in the case of conjoint, students? In the case of conjoint, your xylem and phloem are present on the same radii, not different. For example, there is one sandwich here. All of you see one sandwich now. See this. This is called as same radii. Here we have xylem, phloem, xylem, phloem, xylem, phloem. Can you see? Xylem and phloem are in the same radii. That is called as your conjoint arrangement. It is called as conjoint arrangement. That is found in the case of your, this is found in the case of your leaf and stem mostly. In the case of leaf and stem. Now students, here there is a deviation from here. There is a type of your conjoint here. Understand? There is a type of conjoint here. Two types actually. Two types of conjoint here. Okay. See this, all of you. There are two types here. Okay. Conjoint can be of two types. One is called as collateral. One is called as collateral. Other one is called as bicollateral. Bicollateral. What is the difference? What is the difference? Understand. Difference you understand, all of you. Difference between collateral and bicollateral. In the case of collateral, see, same thing first of all. Same thing is going to be here first of all. Okay. Now the main difference lies in the arrangement. See, middle part is called as what? Middle part is called as the pith. This is the pith. Here also have the pith right here. Okay, pith is here. Now understand, in the case of collateral what is happening. In the case of collateral, xylem is present towards the pith and phloem is present away from the pith. Xylem towards the pith, phloem away from the pith. Xylem towards the pith, phloem away. Xylem on the inside, phloem on the outside. This is called as collateral arrangement. This is called as collateral arrangement. But in the case of bicollateral, what is happening? In the case of bicollateral, there is a sandwich. In the case of bicollateral, there is a sandwich here. That is phloem, xylem, phloem, phloem, xylem, phloem, phloem, xylem, phloem, and phloem, xylem, and phloem. Can you see here? Here there is a sandwich of, here there is a sandwich of xylem. That is called as bicollateral. So radial, conjoint, collateral. All these things you need to remember. Okay, all these things you need to remember. Now students, under your collateral also, there are two categories. There are two categories. All of you need to know this. There are two separate categories under collateral also. What are two categories? Understand. Okay, understand the two categories. Here in the first category, there is a presence of a cambium between xylem and phloem. Can you see here? There is a cambium present between xylem and phloem. This is the xylem, this is the phloem. Here we have again xylem and phloem. Now if there is a cambium present, if there is a cambium present between xylem and phloem, if there is a cambium present between xylem and phloem, it is called as what? Tell me students. Close or open? Tell me students, who can tell me? It is called as what? If cambium present is called as what? It is called as open. It is called as open arrangement. Open arrangement. Now what if there is no cambium? What if there is no cambium? Then it is called as closed arrangement. It is called as what? Closed arrangement. Closed arrangement. This open arrangement is mostly seen in the case of stem. Stem of dicot. Stem. Okay. It is seen in the case of stem of dicot. Stem of dicot. Because if the open arrangement is there, if there is open arrangement, it can undergo secondary growth. It can undergo secondary growth. Dicot stem. Now what about closed arrangement? Closed arrangement is seen in the case of leaf and monocot stem. In the case of monocot stem. So leaf and monocot stem. Leaf and monocot stem is closed. Stem is dicot stem is open. Now root is always going to be radial arrangement. It is going to be radial arrangement. I'm going to move aside, take a picture, draw with me, or do whatever you want, but this should be imprinted in your brain. All this information should be stored in your brain like one Xerox copy. Done? All of you done? Now why did I teach you all of this? Why did I teach you all of this? Because when we have to understand the vascular system, this will come in handy. Okay. For example, right now, all of you write down. Till now, what have we learned? Till now we have learned that we have the epidermis, outermost layer, single layer. It is known as epiblema. Cuticle is absent. Intercellular space is absent. Unicellular root hair. Epidermis. We learned about cortex. We learned about your endodermis. Here we also see this is the Casparian strips. Can you see? Transverse wall tangentially and radially. Tangentially, radially, Casparian strips. Then we learned about pericycle, initiation lateral roots, growth of vascular cambium. Now students, the last point here, the last point which you need to understand with respect to the dicot root is going to be your vascular bundles. It is going to be the vascular bundles. It is going to be vascular bundles. How are they arranged? How are they arranged in vascular bundles here? Now students, you tell me. Look at the diagram. Tell me, how many xylem bundles do you see? 1, 2, 3, 4. Yes or no? Tell me. How, like a clock it is? 1, 2, 3, 4. So can I tell the xylem bundles are technically 2 to 4? Yes or no? 2 to 4. 2 to 4 xylem and phloem bundles. Xylem and phloem bundles. 2 to 4 are there. Okay. Apart from that, we just now learnt. How are they arranged? Are they arranged radially or collateral or conjoint? Tell me students. Are they arranged radially or conjoint or collateral? It is a root. So root is always radial arrangement. Yes or no? Radial arrangement. Always radial arrangement. And it is a patchy. It is always patchy here. Arranged in a patchy-like structure. Now apart from that, there is one more concept is there which I did not teach you right now. That is exarch condition. There is a condition called as exarch and endarch. Exarch means the protoxylem is towards the periphery. Endarch, protoxylem is towards the inside. Okay. So in the case of roots, always we find exarch condition. We find exarch condition in the case of root. So imprint this in your brain. Rx. How do I remember it? I remember it as Rx. Rx bike, you know, right? Rx bike. So Rx bike is nothing but root. Root is always exarch condition. Okay, root is always exarch condition, Rx bike. Okay, exarch condition. Then now students, here between your xylem and phloem, understand? Between xylem and phloem, there is a special type of tissue called as conjunctive tissue here. Here between xylem and phloem, there is a conjunctive tissue here. Okay, there is something called as conjunctive tissue. Conjunctive tissue is present. Conjunctive tissue is present between xylem and phloem. Now apart from that, the only thing we should remember, one second students. Okay. Can you see the middle part? The middle part here is called as pith. It is called as pith. Okay. Now pith here is actually very small. It is said to be inconspicuous. It is very small pith. Also called as inconspicuous, it means it is barely visible. Apart from that, the only thing which you need to remember is the stele here. Stele. Now what is stele, students? What is stele? Stele is nothing but your pericycle, vascular bundles and pith is called as stele. What is stele, students? Stele is nothing but pericycle plus vascular bundles plus the pith is the stele. It is the stele, and that is the end of Dicot root. That is the end of Dicot root. I'll give you a small recap what we learned till now. First, we learned about your epidermis, epiblemma, outermost layer, single layer, also known as epiblemma. Cuticle is absent, ICS is absent, unicellular root hair. Okay. Cortex multi-layer cortex, thin-walled parenchyma, intercellular space is present in some cases, and you're absent in some cases. Then we learned about your endodermis, barrel-shaped cells, single layer, innermost layer of the cortex, biological checkpoint. It has a superior portions in the form of Casparian strips. Pericycle, two main functions made up of parenchyma, few layer, thick-walled. Initiation of lateral roots, ground where we have cambium is present. Then we learned about your vascular bundles, 2 to 4, radial, exarch, conjunctive tissue is present. Pith is small, stele is made up of pericycle plus vascular bundles plus pith. Now students, see this given in NCERT also. Okay, see here, it says sunflower root. Sunflower. So tell me, is sunflower monocot or dicot? Tell me, students, is your sunflower monocot or dicot? Tell me all of you. Tanuja, don't worry, it will happen. It will happen. Plant Kingdom entire one shot I will go in more time. Okay, tell me now students, sunflower is monocot or dicot? We learned that sunflower is your dicot. So in examination they will not give you monocot and dicot, they can give you the examples directly, so be aware of that. Okay, now epiblemma, yes, epiblemma, it has root hair, unicellular root hair. Okay, unicellular root hair is present. Okay, now cortex is thin-walled parenchyma, yes, it is thin-walled parenchyma. Okay, then we learned about your endodermis, yes, endodermis, single layer, barrel-shaped cells. It has Casparian strips in the form of subarine depositions, yes, subarine depositions right here. Okay, thick-walled parenchyma cells made is called as your pericycle. Pericycle has two main functions: initiation of lateral roots and also vascular cambium during secondary growth. Okay, the pith is very small. I told you, pith is very small here. Very small pith. Okay. Between xylem and phloem we have the conjunctive tissue. Tissues. 2 to 4 vascular bundles, xylem and phloem patches. Okay. All the tissues on the inner side of the endodermis, such as your pericycle plus vascular bundles plus the pith, is called as your stele. It is called as your stele, and that is the end of your Dicot root. That is the end of your Dicot root. Did all of you understand? Tell me. So when you write these notes in practice, right, if you read the entire thing like this, paragraph wise, if you read it as paragraph wise, it becomes confusing. Entire thing. But when you write points like this, it becomes very easy to understand, and all of these points you can remember with the help of diagram. Done? Yes, students. See, and which type of questions can you expect here? That also very important, right? Before we go to the question, let's do monocot now. Let's do monocot root. See, dicot root, if you understood, monocot root becomes very easy, because monocot root and dicot root are almost same with small changes. Understand. What are the small changes you understand, students? Okay. In the case of monocot root, usually the epidermis is same. The epidermis is generally same to same. Cortex also is same to same almost. Cortex is same. Pericycle is almost same to same. The only difference in pericycle is going to be the secondary growth is not there. It is monocot root. Apart from that, the main difference comes in your vascular bundles. The difference arises. Okay. Now, what is the difference? Understand. In the case of your dicot root, there were 2 to 4 vascular bundles. All of you remember 2 to 4. But right now, in the case of your monocot, count. All of you count. 1, 2, 3, 4, 5, 6, 7. Read, all of you count with me. 1, 2, 3, 4, 5, 6, 7. So can I say right now, the vascular bundles is more than 6? Vascular bundles is said to be more than 6. More than 6. And such a condition is called as what? Polyarch condition. It's called as what? Polyarch condition. Now, apart from that, this is a root. So, exarch condition. Yes, exarch condition. It is a root. Apart from that, it is a root. So, radial arrangement. Yes, radial arrangement. But students, tell me, this is a monocot. This is a monocot right here. Will it be open or close now? Will it be open or close? Tell me, all of you. Think again. It is monocot. There is no secondary growth, so it should be closed. Should be closed. Should be closed completely. It should be closed completely. Okay. Now, apart from that, the main change will come in the pith. It will come in the pith. Here, the pith is very large and well developed. Pith is very large and well developed. It is well developed pith. That's all. Done. Monocot root done. Monocot root is done. Can you see here? Anatomy of monocot root is similar to dicot root in many aspects. Epidermis, cortex, endodermis, pericycle, almost same. Vascular bundles and pith. As compared to the dicot root, which has a fewer elements, cell bundles, there are usually more than 6. Polyarch condition is present in the case of your monocot. Pith is very large and well developed. Closed. Monocots do not undergo secondary growth. It means it is closed condition. It is closed condition in the case of monocot. Done. Done, students. All of you understood the monocot versus dicot. Root is over. Root is over. Any doubt, anyone, students? I'm looking at the chat right now. Jensly, Nandu, Madhumita, Tanuja. Proud to be plants. Huh? Yes, nice plants. All of you are. All of you are photosynthetic, nice plant, independent, strong, independent plants. All of you are what? Strong, independent plants you are. Strong, independent plants. Because photosynthetic, no? Okay. Understood, all of you? Did you write down the notes? All of you writing the notes, right? If you're not writing the notes, all of you know the notes is available where? All the notes are available on Telegram channel, right? And also on Vedantu NEET English. All the notes are available. Okay, strong independent plants. Yeah, nice plants you are, all of you. Okay, now students, that was your monocot and dicot. Now, which type of question you can expect from here? See, all of you learn the concept, but unless you know how the question can come, all you learn is going to be little difficult to apply. Okay, now see here, question can come in this way. All of you can read the question, I'll read it for you. In a transverse section of monocot root, see, it is a monocot root showing the following internal tissue organization. Okay, arrange the following in the correct sequence starting from the periphery towards the center. From periphery towards the center. Okay, to the center. Tell me, which will be the outermost? Endodermis, pith, epidermis, pericycle, cortex. Tell me, which will be the outermost here now? Which is the outermost? Obviously, you know, your pericycle should be outermost. I'll give you 2 minutes, you sit and do. I'll give you like 10 seconds, sit and do it. Tell me, students, which is the outermost? Outermost is epidermis. So, one is epidermis. Okay. One is epidermis. Then what do we have? Then we have cortex. Yes. Then we have cortex. Okay. Then we have cortex. Okay. Then you tell me, students, what do we have after epidermis? Cortex. Then do we have your endodermis or do we have your pericycle? Endodermis will come first. So 3. Yes. 3. Then we have pericycle. And finally we have the pith. So CEADB, right? CEADB. So all of you understood how the question can be framed here? All of you getting some idea? Yes. Look at this question now. in a transverse section of plant shows polyar condition. See, as soon as you read polyar condition, you should know the answer. As soon as you see polyar condition, you should tell me the answer. It is radial and X arch. If it is radial and X arch, it is supposed to be what? Root only. Radial arrangement is root. X arch is root. Polyarc is monocot or dicot? Polyarc is monocot or dicot? Tell me now. Polyarc is monocot. Yes or no? It is monocot root. Monocot root. So you can answer the questions very easily if you know the exact points which you need to match. So there are some points in your head now. Match the following points to the question. Answer is yours. Answer is yours. Okay. This is a NEET 2023 question. See this question, all of you. Initiation of lateral roots and vascular cambium during secondary growth takes place in. It is a NEET 2022 question. Initiation of lateral roots. Two main functions we learned. That is your pericycle. Yes or no? In pericycle we learned that initiation of lateral roots as well as your secondary growth is taking place. Secondary growth is taking place. Tell me, is anatomy very fun and easy also? Anatomy is fun and very easy to do. Anatomy. Okay. Now, look at this question now. In dicot root, oh, this is your removed part. Don't worry about this. This is your secondary growth. Okay. See this question now. Casparian strips occur in. That is, subarine depositions. Subarine depositions happens where? In the case of epidermis, pericycle, cortex, or endodermis. This is a question from NEET 2018. It's an old question, but still applicable. Very question, easy answer. Casparian strips is found in your barrel-shaped cells, endodermis. Tell me, endodermis, done. Okay, so that was the different questions we can expect. Those were the different questions you can expect in your examinations, the type of questions. Okay, I'll ask you one more question. Okay. In which of the following monocot or dicot root, where do you find closed vascular bundles? Go. See, they'll give you multiple options. It is X arch condition, it is said to be radial arrangement, but it is closed. It is closed. Cambium is absent. Is it now monocot root or dicot root? Tell me now. Is it monocot root or dicot root? You will tell me it is monocot root. Why? Closed condition. Because of closed condition. Okay. So such questions can come. Such questions can come in your examinations. Okay. Now students, with that being said, can we start dicot stem now? Stem part. Can we start? Can we start stem now, students? Students, all of you quickly smash the like button. Smash the like button. And students, do you realize, do you know why did I take up this topic? Because 1 week back, I got a comment. 1 week back, I got a comment on one of my videos telling that, sir, monocot versus dicot is very difficult for me to understand. Can we have a separate session for it? And similarly, students, I got a request from all of you itself in one of the comment section telling that, sir, I cannot tell difference between C2 cycle, C3 cycle, C4 cycle. Now the question is, do you want a session on that? Do you want a small session on what is C2 cycle, what is C3 cycle, and what is C4 cycle? So we can keep these small sessions for concept building, and you cannot go wrong here. Okay. Now let's come to dicot stem now. Dicot stem. Let me drink some water. Show some energy in the chat, all of you. Now, dicot stem. Okay. Now, students, if you look at the diagram and tell me which is the outermost layer right now, tell me in the chat, all of you. I'll give you 1 minute. I'll give you 20 seconds, not 1 minute. I'll give you 20 seconds. Tell me which is the outermost layer in the case of dicot stem. Do you see in the diagram? Don't look at my face. Don't look at anything. Just look at the diagram and tell me which is the outermost layer right now. Can you see here? The outermost layer right here is called as your epidermis again. It is called as epidermis right here. It is called as your epidermis right here. Okay. So can I write it outermost layer? Yes, outermost layer. Outermost layer. Outermost layer right here. Can I tell it is Productive in function. Yes, sir, it is productive in function. Productive in function. Okay, productive in function. Now, students, do you see it as a single layer or multiple layers? Tell me. Is it present as single layer or multiple layers? Single layer or multiple layer? Tell me. You tell me, sir, single layer. It is present as a single layer. Okay. Single layer present right here. Single layer. It is made up of parenchyma. Parenchyma. It is made up of parenchyma right here. Okay. Usually this parenchyma has no chloroplast. Chloroplast is absent because no function. No chloroplast. No chloroplast right here. No chloroplast right here. Okay. Now apart from that, do you see any kind of appendages here? Do you see any form of appendages here, right here? Can you see? When antennas are here, 3 antennas. Okay. Can all of you see? Yes, you can see. Okay. So these appendages are called as what? Trichomes. Called as trichomes. They're called as what? Trichomes. They're called as your trichomes, which help in multiple function, secretory functions, increase in surface area, all of that is there. Trichome is present. Apart from that, do you see anywhere intercellular spaces? Tell me, yes or no? Any form of intercellular space in a single layer? No. Intercellular space is completely absent here. Apart from that, in this particular stem, it is a stem on the outside, dicot. So the cuticle may be present or may not be present. The cuticle may be present or may not be present. The same goes for stomata. The stomata can be present or it cannot be present. The same goes for stomata also. Can be present in some cases, cannot be present in some cases. That is your epidermis. So what is the difference between trichomes and root hair? Trichomes are present in the case of stem, root hair is present in the case of root, and trichomes are usually multicellular and branched. Okay. Done. That is your trichomes. Right here. Okay. Done. Epidermis done, all of you. Epidermis is done. Okay. Epidermis is done. Now, students, the next thing you need to understand here is the ground tissue system. Next thing is called as your ground tissue system. Okay. Let me draw one. I'll take a separate slide. Okay. We'll take a separate slide only. Okay. Next thing is actually called as your ground tissue system. Write down this way. Ground tissue system. Ground tissue system. This ground tissue system is divided into 3 different parts, 3 different subzones. 3 different subzones. 3 different subzones are here. 1 second. From here. Ground tissue system. It has 3 subzones. Okay. The first one is called as your hypodermis. The middle one is called as cortical layer. And the innermost of the cortex is always endodermis. It is always endodermis. See this, all of you. Here we have, can you see? This is the entire thing is called as your cortical layer. This entire thing is called as your cortical layer. This entire thing is called as, so the entire thing is called as ground tissue system. This entire from here. This entire chunk is called as ground tissue system. The outermost layer, this is the hypodermis. Then we have your cortical layer. And then we have the innermost layer, that is endodermis. Now students, tell me, what is hypodermis made up of? Look at it and tell me. What is hypodermis made up of? Right here. It says collenchyma. Yes or no? Collenchyma. So hypodermis is made up of what? Hypodermis is made up of 𝐂𝐤. It is made up of 𝐂𝐤. It is made up of 𝐂𝐤. Now here, do you see it as a single layer or multi-layer? Do you see it as single layer or multi-layer? All of you see this? See the 𝐂𝐤? In 𝐂𝐤, do you see it's not just present as a single layer, it is present as many layers here. Yes, it is many layers. Okay, it's generally few layers. It is few layers, and at the same time, interstellar space is generally absent. Now, since it is 𝐂𝐤, tell me, students, it can provide mechanical strength. It can provide mechanical strength and also be photosynthetic in many cases. Photosynthetic in many cases. Mechanical strength and photosynthetic in many cases. That is your hypodermis. Okay, hypodermis. Now, students, the next layer is called as your cortical layer. Can you see this? Most of this part is called as your cortical layer. The second layer is cortical layer. Now, cortical layer, if you look at the cortical layer, it is made up of parenchyma. First of all, it is made up of parenchyma. From here we can tell. Made up of parenchyma. Here it is thin-walled round parenchyma cells. Thin-walled round parenchyma cells. It is made up of what? Made up of thin-walled parenchyma cells. Parenchyma cells. Thin-walled parenchyma cells, and interstellar space is present generally here. Parenchyma, right? It is generally present here. Okay. Now, students, your innermost layer is called as your innermost layer of your ground tissue system is called as your endodermis. You see here, it is called as your endodermis. Endodermis. Now, there is a speciality of endodermis now. In this endodermis, there is presence of starch grains here. There is a presence of starch grains. In the endodermis, there is the presence of starch grains. Starch grains. Now, all the starch grains are present in the form of a layer. In the form of a layer, and the entire layer is called as what? Entire layer is called as starch sheet. Starch sheet. That is your endodermis. Students, are you writing down all this? Is everyone writing down? The ground tissue system is divided into three separate categories. Called as your hypodermis, cortical layer, and endodermis. Right here, this much. Hypodermis, cortical layer, endodermis. This is your entire ground tissue system of your dicot stem. Okay. Now the speciality here is endodermis is starch sheath, hypodermis is 𝐂𝐤, cortical layer is parenchyma. That's all. Nothing more than this you need to remember. For your cortical, for your entire epidermis. So your entire ground tissue system. Now, students, you tell me, all of you in the chat, which is the next layer? Do you see here? Which is the next layer do you see here? Tell me. Which next layer do you see there? Tell me, students. All of you are aware, this is called as full moon. Full moon. Okay, this is full moon. Okay, Purnima. Now, what if there is half moon? Full moon is called as lunar. Can I tell half moon can be called as your semi-lunar? Okay. Now, if you look at the shape of your pericycle, it is semi-lunar shape. So, pericycle. The first point here of pericycle is going to be your first point about your pericycle is going to be the location. Students, your pericycle is present on the inner side of endodermis. And away from the phloem. Inner side of endodermis and above phloem. Can you see? It is above the phloem. Can you see here? It is above the phloem. This is the phloem right here. This is the phloem right here. So your pericycle is present above the phloem and inner to the endodermis. Inner to the endodermis. It is semilunar shape. Semilunar shape. And apart from semilunar shape, if you look at this section right here, can you see the pericycle is wavy? All of you can see here. Pericycle is nicely wavy. Do you see that? Pericycle is nicely wavy there. Yes. So can I tell the pericycle is said to be wavy? Wavy. Pericycle is wavy. Wavy. Okay, wavy pericycle. Apart from that, the pericycle is made up of sclerenchyma. It is made up of sclerenchyma. Okay. It is made up of sclerenchyma, and that is the end about your pericycle. That is the entire thing about your pericycle. Entire thing about your pericycle. So it is inner side of the endodermis and above the phloem, semilunar shape, semilunar shape. Okay, wavy in nature, and also it is made up of sclerenchyma. It is made up of sclerenchyma. Now, students, do you want to know one extra thing here? Do you want to know one extra information? Because in the members of cucurbita, it is made of sclerenchyma completely. But at the same time, in the case of your what is that? In the case of your sunflower, the pericycle is actually made up of a mixture. It is heterogeneous in origin. It is like the entire pericycle is made up of your sclerenchyma, parenchyma. That is in the case of your sunflower family. Not given in NCERT, but extra information as a botanist, I can tell you. Okay, so that is your pericycle. That is your entire pericycle. Now, students, pericycle is over. Now, what is the inner to the pericycle? What is present inner to the pericycle right now? Inner to the pericycle. Inner to the pericycle is the entire vascular bundles. Entire vascular bundles present right here. Can you see? Yes. Inner to the vascular bundles, we have the inner to the pericycle, we have the vascular bundles. Vascular bundles. Okay. Now, first thing you need to know about vascular bundles here is, if you look at the entire vascular bundles, doesn't this look like a ring arrangement? Yes or no? Tell me, students. Doesn't it look like a ring arrangement? Tell me, all of you. One, you can take this and wear it in your hand. Yes or no? One ring arrangement is there? Yes, there is a ring arrangement. There is a ring arrangement. Complete ring arrangement can be seen here. Complete ring arrangement. Now, since it is a stem now, stem, not root, will it be conjoint or will it be radial again? Tell me. See, radial in the case of your root, radial in the case of root, but in the case of stem, it is supposed to be conjoint. Xylem and phloem are present together. Can you see here? In this diagram, if you see, here also we can see. Here we have phloem, here we have xylem. Xylem and phloem are present on same radii, xylem and phloem. So can I call this arrangement as conjoint? Conjoint. Yes. They are present as conjoint. Now xylem is present towards the pith. Xylem is present towards the pith, phloem away from the pith. So can I call it collateral? Collateral. I can call it as collateral arrangement. I can call it as collateral arrangement. Since it is a stem, will it be endark or exarch? It is going to be endark. It is going to be endark arrangement. Endark arrangement. So all of these points, each individual point right here is a PYQ question. PYQ question. And all of this will come with experience. Just because you're watching my lecture for the first time, it doesn't mean that you'll remember everything altogether. This lecture is providing you a particular guidance how you should approach anatomy, how you can study anatomy, how you can understand anatomy. Okay, so you have to learn this, write down this and learn later on. Okay, so endark arrangement, collateral. Now students, tell me, it is a stem. It is a stem. Will it be open or close? Tell me, students, will it be open or close now? Obviously, it is going to be closed arrangement or open arrangement. Tell me. See, it is a stem. It is dicot. It is showing secondary growth. So can I call it as open arrangement? Open arrangement. Open arrangement. That is your vascular bundles. Okay. Now, students, Here you need to understand between xylem and phloem, between your vascular bundle, not xylem and phloem. This is one bundle. This is one bundle. Between two vascular bundles, there is a medullary race coming out here. Between xylem and phloem, between your vascular bundles, there is medullary race coming like this everywhere. Medullary race here. Between your vascular bundles, there is medullary race, which undergoes dedifferentiation to produce your secondary meristem later on. Okay. So here we have what? Between vascular bundles, what we find, students? We find something called as medullary race. We find something called as medullary race. Medullary race are present here. Okay, medullary race. Now, what is the function of medullary race, you might ask me, sir? Function of medullary race is that it helps in conduction of water, conduction of different types of minerals and organic solvents and everything, organic acids. So helps in conduction. Medullary race help in conduction. Okay, so that is your entire vascular bundles. Only part remaining is the pith. Now see, we're coming from top to bottom, so pith is remaining. What is pith? Pith is very large here. The pith is very large. Parenchyma, intercellular space is present. That's all. The pith is very large. Intercellular space is present here. It is made up of parenchyma. Parenchyma, which is round shaped cells. That's all. Now students, see this. All of you ready for revision? I'll revise this only once for you, then we will see the NCERT revision. First, we learned about your dicot stem as your epidermis, outermost layer, productive in function, single layer, parenchyma, no chloroplast, trichomes are present here, intercellular space is absent, cuticle may be present, may not be present, stomata may be present, may not be present. Then we learned about your ground tissue system. The ground tissue system is mainly divided into three parts: hypodermis, cortical layer, and endodermis. Hypodermis, collenchyma, parenchyma, starch grains. Few layer, no intercellular space, mechanical strength, and photosynthesis. Collenchyma, no. Then we learned about your parenchyma, intercellular space is present in cortical layer. Endodermis, starch grains as form of starch sheet. Okay. Pericycle, it is present inner to the endodermis, above the phloem. It is semi-lunar in shape and also it is wavy in nature. It is made up of sclerenchyma, red color, sclerenchyma, remember it, sclerenchyma. Then we see the vascular bundles, entire vascular bundles, ring arrangement. It is conjoined, collateral, end arc and open. Between the vascular bundles, there is medullary race. Between the vascular bundles. Between the vascular bundles, there is medullary race. Okay. Then there is pith, very large pith. Intercellular space is present. It is made up of parenchyma. Done. See this NCERT now. See all of you. Outermost layer of the stem, outermost layer. Thin cuticle, it can be present. Okay. Trichomes, yes, trichomes are present, epidermis. Then we have the cells arranged in multiple layers between the epidermis and the pericycle constitute the cortex. The cortex is divided into three subzones. Outer is called as what? Hypodermis. Middle is called as cortical layer and inner is called as endodermis. Done. Can you see? Collenchyma, hypodermis. Cortical layer, parenchyma. Okay. Endodermis has what? Endodermis has starch sheet, rich in starch grains, as in referred to as starch sheet in the case of endodermis. Pericycle. Okay. Pericycle present on the inner side of the endodermis and above the phloem. Made up of sclerenchyma, semi-lunar shape. Done. In between the vascular bundles, there is something called as conjunctive tissues. Vascular bundles are few layers of radially placed parenchyma cells which constitute the medullary race. There is medullary race. Conjunctive tissue in the root. Okay. Large amount of vascular bundles are arranged in a ring arrangement. Ring arrangement is seen there. Okay. or every single point is same to same, which we have learned right now. Same to same. You need to remember, don't read the entire paragraph. I repeat again, do not read the entire paragraph. Read the points, you're good to go. Learn the points from the diagram, you're good to go. You can solve any question from anatomy. Any question from anatomy. Okay. Lastly, we have the pith. Okay. Now, if you learn the dicot stem, monocot stem is very easy. Monocot stem is very easy. Okay, very easy. Now, if you remember, students, I'll show you after this. Monocot stem is very, very easy to understand if you understood dicot now. Why? Because in the case of your monocot stem, epidermis is same. Epidermis is going to be the same to same. That is same to same almost as dicot. The only difference is here you'll have barrel shaped cells, not given in NCERT also. Not given in NCERT. So I'll not tell you extra that much. Okay. The one thing you need to remember here is cuticle is present. It is very important. Cuticle is present here. Okay, cuticle is present. Generally, cuticle is present. Okay. Cuticle is present because it prevents water loss and everything. Prevents water loss. Okay. Now, students, understand the hypodermis. See, all of you know. Now, all of you tell me, in the case of dicot, stem, what was hypodermis made up of? Tell me, all of you. What was hypodermis made up of? In the case of dicot, hypodermis is made up of 𝐂𝐡𝐞𝐤. But students, if you remember, 𝐂𝐡𝐞𝐤 is completely absent in the case of monocots. Now tell me, students, what is hypodermis made up of here then? If there is no 𝐂𝐡𝐞𝐤, there has to be a substitute for it. Yes, there is a substitute. Here it is made up of sclerenchyma. Hypodermis is made up of sclerenchyma here. It is made up of sclerenchyma. It is generally 2 to 3 layers. 2 to 3 layer. Okay. So 𝐂𝐡𝐞𝐤, here it is present sclerenchyma. 𝐂𝐡𝐞𝐤 is absent. 𝐂𝐡𝐞𝐤 is absent in the case of monocot. It is absent in the case of monocot. 𝐂𝐡𝐞𝐤. Very, very, very important line this is. Many mistakes can happen in this small fraction of things. And remember, students, at the same time, 𝐂𝐡𝐞𝐤 will never be present in the case of root. The only place you can find 𝐂𝐡𝐞𝐤 in a plant, entire plant, is mainly the stem of dicot. Okay, and also in the petiole of a leaf. You'll find 𝐂𝐡𝐞𝐤. Okay, because it produces elasticity. It gives you elasticity. Okay. Now, if you look at the ground tissue system, very funny it is here. Okay. If you look at the ground tissue system, do you see anything here? Do you see anything, something called as hypodermis? Do you see anything called as your hypodermis is there? Do you see anywhere cortical layer, endodermis, pericycle, pith? Do you see anywhere, students? Do you see anywhere? There's something called as your pith, pericycle, cortical layer. Nothing is present here. Everything is not differentiated here. So the ground tissue is not differentiated. The ground tissue is not differentiated here. There is no differentiation here. It is generally made up of parenchyma. That's all you should remember. Generally made up of parenchyma with interstellar spaces generally present here. There is no cortex, there is no pith, there is no pericycle, nothing is there. It is not differentiated, just made of parenchyma, and small spaces are there. Okay. But students, what is interesting to note here is the vascular bundles. The vascular bundles is very interesting here. Why is it interesting? Understand. If you look at the vascular bundles, see here, students. This is your vascular bundles. Can you see? Vascular bundles. Towards the periphery, do you see a lot in number? Yes or no? Towards the outside. In outside or periphery, they are more in number, and also small size. Okay, small size and more in number. But as we come towards the inside, do you see they're increasing in size? Increasing in size? Yes or no? They're increasing in size and the number is decreasing now. Yes. So towards the inside, so less in number and bigger size. That is very interesting to note here. Apart from that, since it is a monocot stem, since it is a monocot stem, will it be radial arrangement or will it be conjoint and collateral? Tell me. Since it is a stem, will it be radial arrangement or will it be a conjoint and collateral? Conjoint and collateral. Yes or no? It will be conjoint and collateral. Conjoint and collateral. Okay. Now, since it is monocot, will it be open or close? Tell me. Open or close? It is a stem. It is monocot right now. Monocot means closed. Monocot means completely closed here. Monocot means completely closed. Since it is a stem, it is always end arc. It's always end arc. Always root is exarch, stem is end arc here. Stem is end arc. Now, students, there is one point which I'll tell you right now, which has been as a PYQ many times. One point I'll tell you right now, which has been asked as a many times as a PYQ. That is, phloem parenchyma is absent here. What is absent? Phloem parenchyma is absent. Phloem parenchyma is absent. Phloem parenchyma is completely absent here. Absent. Now, students, if you look at the vascular bundles, for example, this is a vascular bundle. This is a vascular bundle right here. Xylem and phloem. There is no cambium. Around the vascular bundle, there is a sheet. Around the vascular bundle, there is something called as bundle sheet. Bundle sheet is there. What is there? Bundle sheet. And this bundle sheet is made up of sclerenchyma here. Okay. Now, apart from that, students, I'll take you back to this diagram. The first diagram which I showed you here. Can you see here? This is a monocot stem. All of you can see right now, it looks like one skull face. Can you see on the periphery we have more near number and also small size. As we come towards the center, it is larger and few in number. This is called as your vascular bundles. Okay. Now, in this vascular bundles, do you see this small gaps here? All of you, do you see this? On skull face, eyes and nose. Can you see all of this? All of you see this small round structures? Can you see? All of you can see this round structures here, which looks like eyes. On skull shape it is. These eyes are nothing but water cavities. They're called as what? Water cavities. And this is how you identify actually monocot. When you look at the monocot, look like on skull shape it is. Skull shape. Okay. So the presence of water cavities. There's presence of water cavities here. What is present? Water cavities are present inside the vascular bundles. Water cavities are present. Okay. So very interesting it is. So what we learn right now, students? We learned that monocot stem, epidermis cuticle, hypodermis sclerenchyma, ground tissue system is not differentiated. Parenchyma shows intercellular spaces. Outside there is more, inside there is less. Vascular bundles, conjoint, close, collateral, end arc. Phloem parenchyma is absent. Bundle sheet has sclerenchyma and water cavities are present. Same thing is given in your NCERT. So monocot stem has sclerenchymatous, hypodermis. Why? Because collenchyma is absent. Scattered vascular bundles. Surrounded by a sclerenchyma, bundle sheet. Yes. Large conspicuous parenchyma, ground tissue. Vascular bundles are conjoint and closed, even collateral also. Okay. This is the thing. Vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent. The most important point. Phloem parenchyma is generally absent here. Water containing cavities. Can you see? Water containing cavities are also present in the vascular bundles, which gives us the characteristics, that skull shape. The skull shape, no, that is because of your water cavities. Water cavities. That is the entire thing is called as, entire thing is for your monocot stem. Tell me the answer to this question now. Tell me the answer to this question, all of you. The question is very simple. A major characteristics of monocot root is. Now this was a very scam question. This was a controversial question. Okay, this was a very controversial question because let me tell you, these two answers were controversial. Can you see, all of you? Scattered vascular bundles are technically correct to some extent, but the main thing which you need to focus here, the most probable answer. There's something called as most probable answer now. Vasculature without cambium. It means what? It means closed condition. Closed condition. Yes or no? Since it is a monocot root. Oh, this is not. One second. No, this is in your. This is in the case of monocot stem. This is monocot stem. Monocot stem. Okay, this is monocot root. Since it is monocot root, the answer is very clear now. Okay. Monocot without cambium. Next question. The okay, this one it's old NCERT students. This they will not ask you this anymore. But let's see if you can answer this question, students. Can anyone answer this? Interfascicular cambium arise from. They don't usually ask this now anymore. They don't ask this anymore, but let's see if you can answer this question. Remember, between xylem and phloem, there was something, right? I'll just the clue. Between xylem and phloem, there was something in the middle, right? Medullary ray. The medullary ray undergoes dedifferentiation to produce interfascicular cambium. Okay, medullary ray. Water containing cavities in vascular bundles are found in. Students, see this? See, we know sunflower is what? Sunflower is going to be dicot. Cycas and pinus both are gymnosperms. Gymnosperms gone. So maize is what? Maize is a monocot. Maize is a monocot, and water containing cavities are present in the case of monocot. So answer you see. Answer you see. 2 is the answer. Okay. So such questions can come. But they can ask you questions based on the examples. Based on the examples. So be very careful there. Okay. Priyanka, no. Priyanka, not sunflower. Sunflower is a dicot. Sunflower is a dicot. See, that's why I made you write some examples in the starting. In the starting, I made you write some examples. Can you see here? Sunflower is a dicot. Rose, legume, all this is dicot. While your grasses, lily, wheat, rice, maize, orchids, all of these are monocots. All of these are monocots. Okay. Now, students, with that question, we come to the leaf part now. We come to the leaf. Okay. Last part for today's class. It'll take me next 15-20 minutes. Ultimate focus. Can we start? Last 15 minutes, 15 to 20 minutes. Ultimate focus. Can we finish the leaf now? Can we finish the leaf? Can we start the leaf part now? Ultimate focus. I want ultimate focus right now. Listen to what I'm saying and you will enjoy the leaf part also. It's very, very easy to understand leaf also. Okay. Let's start with the leaf right now. Let's start with the leaf now, students. Let's start with the leaf right now. Okay. Now, let me draw a simple leaf for you. Let me draw a simple leaf. This is a dicot leaf. This is a dicot leaf. Remember, why dicot leaf? Because of reticular divination. Because of reticular divination, this right here is a dicot leaf. Okay. Now your leaf usually has two different epidermis, upper epidermis and lower epidermis. Two epidermis are present here. This is called as your lower epidermis. This is called as your lower epidermis. Lower epidermis. This is called as upper epidermis. Upper epidermis. Upper epidermis. Students understand, lower epidermis is also called as dorsal side. Dorsal side, and also called as, see, lower is always referred to as back, lower back side. So it's also called as your abaxial side. Abaxial side. Abaxial side. Okay. So if this is dorsal, this is going to be ventral side. Ventral, also called as adaxial side. Adaxial side. So lower, upper, lower, upper. Now students, we need to understand something called as your dicot leaf. Dicot leaf. Okay. A dicot leaf is also called as, can anyone tell me what is the other name of dicot leaf? Quickly tell me in the chat. I'll give you 20 seconds. Is it called as dorsiventral or is it called as isobilateral? Tell me students. What is the other name of dicot leaf? Is it called as dorsiventral or is it called as dorsiventral or is it called as your isobilateral? Anyone? Anyone? It is called as dorsiventral leaf. It is also called as dorsiventral leaf. Dorsiventral leaf. Now why? Why do we call it as dorsiventral leaf? Why? The question is why. Now students, I just now told you, we have the dorsal side and we have the ventral side. We have the dorsal side and we have the ventral side. Upper epidermis, no, this is upper, right? Upper, not given here, okay. So this is ventral side, dorsal side. Dorsiventral. Dorsiventral means in both the sides, we have a different types of mesophyll cell. Look at the diagram, look at the diagram. This is the dorsal side, ventral side. In ventral side, see, the upper epidermis, usually ventral side, we have the palisade mesophyll. In ventral side, we have the palisade mesophyll, and in the dorsal side, we have the spongy mesophyll. So reason it is called as dorsiventral means dorsal side and ventral side, there is a different types of mesophyll cell. Dorsiventral leaf. Okay, dorsiventral leaf. I hope you can remember that. Okay, dorsiventral leaf. Now students understand, this entire leaf is divided into three main parts. Entire leaf is divided into three main parts. First we have the epidermis, then we have the cortex, and then we have the vascular bundles. Now which is the outermost layer, students? The outermost layer is called as epidermis again. The outermost layer is called as what? Epidermis again. Epidermis. Now here epidermis is of two types. We have the upper epidermis and we have the lower epidermis. Two different types of epidermis is present here. Two different types of epidermis is present here. Clear, all of you? Clear? Upper epidermis and lower epidermis. Now students, I'll give you a scenario. I'll give you a small scenario. See this. This is the stem, nodal region. In the nodal region, we have a leaf here. Okay. We have a leaf in the nodal region. Let's say we have a leaf here. Leaf here. Okay. Now here, from here, the sunlight is coming. Sunlight is falling on the leaf. Now students, tell me, in which side do you have more stomata? Tell me, in which side do we have more stomata? If sunlight is coming from the top, obviously we need to avoid the stomata there because of transpiration loss. Yes or no? Yes or no? So tell me, which side do you think will have more stomata? Upper side or lower side? Tell me students, which side do you think will have more stomata? Obviously on the lower side. Yes or no? On the lower side we can find more stomata. Yes or no? More stomata. On the lower side, because it is protected from sunlight. This lower side is protected from the sunlight, so we find more stomata on the lower side. And usually in some cases, in epidermis, there is no stomata only. There is no stomata on the upper side also. That is also the case. some cases. Now students, the leaf is exposed to sun. Does it need a moisturizer? Tell me. Does it need a sunscreen? Does it need a sunscreen? Obviously, it needs some nice cream. It needs a shiny layer there called as cuticle layer. Yes or no? Yes or no? It needs a cuticle layer here. Cuticle layer. It needs a cuticle layer is present here. Cuticle layer is present here. That is the epidermis. That is the epidermis. But if you look at the cortex now, look at the cortex. All of you look at the cortex now. The cortex is made up of something called as mesophyll cell. It is made up of something called as mesophyll cell. Can you see here? It is made up of something called as mesophyll cell. Now there are two types of mesophyll here. There are two types of mesophyll here. Can you see all of you? One is present on the top, one is present on the bottom. The one which is present on the top is called as palisade. It is called as what? Palisade mesophyll. It is called as palisade mesophyll. One which is present on the bottom is called as spongy. It is called as spongy. So here we have palisade, here we have spongy. Palisade and spongy. Okay. Now students, if you look at the diagram of palisade, you can tell me how is the arrangement. See the diagram of palisade, all of you. If you look at the diagram of palisade, can you see they're arranged vertically and parallel to each other? They are arranged vertically and parallel to each other. Parallel to each other. Yes, parallel to each other. But at the same time, how is a spongy arranged? Spongy is round and oval in shape. Yes, round and oval. Round and oval. And also it is very loosely arranged. Yes or no? Loosely arranged. Yes or no? Can you see here? It is completely loosely arranged, entire thing. And there is also, because of loose arrangement, there is something called as air cavities. Can you see here? Air cavities are present here. Air cavities are present here. Presence of air cavities. Presence of air cavities are also present here. Air cavities are also present here. Done. Air cavities are also present here. So cortex is mesophyll. Mesophyll is divided into palisade and spongy. Lower we have monostomata. Lower we have stomata. Okay. There is abaxial, adaxial. Adaxial upper side. Abaxial is the lower side. Abaxial is the lower side. Now students, if you look at the bundle sheet, can you see around the vascular bundles? There is a bundle sheet here. Yes, can you see around the vascular bundle, around the xylem and phloem. Around the xylem and phloem, there is a separate layer that is called as bundle sheet, which is made up of parenchyma. Parenchyma here. Okay, made up of parenchyma. Now one more thing you need to understand about vascular bundles here is if you look at this leaf, the size of the veins is equal to the size of the vascular bundles. What do I mean by that? Understand. Here we have large vein equals to large vascular bundle. Okay. Here we have small vein equals to small vascular bundle. So size of the vein is equal to the size of the vascular bundles. Okay. Now, since it's a dicot, we are talking about dicot here. It will usually show reticulate venation. Reticulate venation. Okay. So secondary row is deleted. Okay, be happy. Leaf is very easy to understand. There's nothing difficulty here. That is the end of dicot leaf. I can show you. See here. It is called as dorsiventral. All of you know why. Okay. Epidermis, two epidermis covered by upper epidermis, lower epidermis. Okay. Cuticle is present. Yes, cuticle is present. Sunscreen we need. Okay. See, lower side even upper side may lack stomata because of sunlight. They have mesophyll cells, palisade and spongy. Okay. See, palisade is parenchyma is elongated cells which are arranged vertically and parallel to each other. Other than spongy is oval and round and loosely arranged. Spongy. Okay. Vascular bundles includes vascular bundles which can be seen in the form of veins and midribs. Veins and midribs. There is a bundle sheet which is made up of parenchyma. And that's it. That's it. Now if you see the monocot, monocot is very simple. Now I'll show you one. Here monocot, I'll tell you students, monocot again epidermis is same. Epidermis is same. Generally same. But stomata will change. Stomata will change. I'll tell you why. See this. This is the typical monocot leaf. Monocot leaves are generally much more parallel to the ground. They're much more parallel to the ground. Can you see? They're much more parallel to the ground. Okay. Now in such cases, the sunlight doesn't play much role. Sunlight is almost hitting equally. Sunlight is almost becoming equally round. Equally. Okay. Sunlight is hitting almost equally right now. And with that, can I say stomata is generally equal on both sides. Equal on both sides. Equal on both sides. Okay. Now what is protoxylem and metaxylem? Protoxylem and metaxylem is protoxylem is first formed xylem. Metaxylem is present later on, produced later on. Protoxylem is first formed xylem. Metaxylem is later formed xylem. Okay. Not your secondary xylem. Not the secondary xylem. It's a type of primary xylem only. Okay. Type of your primary xylem itself. Now since it is a monocot, I will have your parallel arrangement of veins. Parallel arrangement. Yes, parallel arrangement will be here. Now apart from that, if there is parallel arrangement, there is nothing called as larger midrib, small vein, bigger vein, small vein, nothing like that. So usually since it is parallel arrangement, the size of vascular bundle is equal, generally equal, unlike your dicot. Now apart from that, students, there is nothing called as your palisade, there is nothing called as your spongy. That is why it is called as what? That is why it is called as isobilateral. Iso means same. Isobilateral. The meaning of iso means what? The meaning of iso means same, bi means both, lateral sides. So same type of mesophyll is present on both the sides. That is basically your mesophyll. There is nothing called as your spongy. There is nothing called as your palisade. Same type is present on both the sides. That is why it's called as isobilateral. Iso means same on both the sides. Isobilateral. Okay. Xylem and phloem, mesophyll is there. You can find air cavities here. Stomata is almost equal on both the sides. Stomata is equal on both the sides. Okay. Clear, students? The only point which I have to tell you is something called as bully. Called as a bulliform cells. Bulliform cells. Now what is this bulliform cells? Understand, students. All of you can see this epidermis. We can see the epidermis. Some of the epidermis become large, colorless and empty. What happens? Some of the epidermal cells become what? They become large, colorless and empty. Very sad, no? Very sad, yes. Large, colorless and empty. If they become large, colorless and empty, they can store what? They can store water. Okay. So when there is water is present, the entire leaf looks something like this. Can you see? The entire leaf look like this. And bulliform cells has water. It is turgid. As soon as all the water is removed, if you remove the entire water content inside the bulliform cells, the leaf will show. This is the leaf. It will show curling. Can you see? The leaf will start to curve inwards. This is called as curling of leaf. Not this way. It is generally this way. Curling of leaf. Curling of leaf is observed in the case of your monocot. Because if bulliform cells lose all the water, they will show curling. Okay. Large, empty, colorless cells. Okay. See, isobilateral is similar to that of dorsiventral, but there are small changes. What are small changes? Stomata is present on both the surface and epidermis. Stomata is present on both the sides equally. There is no differentiation between palisade and spongy. Okay. There is large empty colorless cell called as bulliform cells which can hold water. Parallel venation is monocot leaves is reflected in the near similar size of the vascular bundles. Yes or no? I told you here. Will I tell you? See, size of vascular bundle is equal because of parallel arrangement. Okay. And see the vertical section of the leaf. This is a question. All of you try answering this. All of you on the board. You have to tell me, choose the correct answers from the options given. Which is the right answer? How many questions are there? Many questions are there. Okay. See, in roots, xylem and phloem in vascular bundles are arranged in alternating manner along different radii. Yes or no? Radial arrangement, roots. True or false? Tell me. In roots, it is radial arrangement. Conjoined closed vascular bundles do not possess cambium. So it is closed, no cambium. Correct. In open vascular bundles, cambium is present between xylem and phloem. Yes. Open means cambium is present. Okay. Vascular bundles of dicot stem possess endark protoxylem. See, it is a stem, so endark makes sense. In monocot root, usually there is, see, it's a root monocot. Polyarch condition, correct. So what is the answer? Tell me what is the answer now? Everything makes sense, right? Everything makes sense right now. Ravindrani, I saw the answer. Don't hide to hide it. Okay, I can see answers. So all of you are able to see the answers we can get from the concepts we learned today. All right. Oh, nice. Now you see the answer. Okay. See this question. Large empty colorless cells of adaxial epidermis along the veins of the grass is called as bulliform cells. Yes or no? Called as bulliform cells. They can ask you a question like this also. It's NEET 2024 this year question. Bulliform cells are responsible for what? They're responsible for the, see, inward curling of leaves. Yes, curling of leaves. Now, this is your homework question. In the comments. If you don't put it, I'll stop giving homework. If you don't add in the comments, I'll stop giving the homework only. This is the homework. Okay. I'll give you a small clue before I go. Let me tell you. Large number of scattered vascular bundles. Oh, this is the clue right here. Scattered vascular bundles. Okay. See, it is supposed to be closed. It means it has to be monocot. Closed. This also is a major clue. Phloem parenchyma is absent is also a major clue. So this is a question from NEET 2020. Let's see if you can solve it. Okay, this is a homework question. Okay. So students, with that, today's lecture is over. Done. Okay. So let me know in the comment section which type of sessions you read. See, this entire session today was based on one small comment of a student. See, I was looking at the comments. Generally, I look at the comments after 1 week or something, or after 2 to 3 days. Okay. When I was looking at the comments, I saw one of the students saying, "Sir, can you teach me what is the difference between monocot and dicot?" Simple question he asked me. So, can you tell me the difference between monocot and dicot in detail in one lecture? And this is a lecture for it. So, let me know in the comment section which type of lectures you want from my end. And let's see if I'll pick up a few lectures from the comments and I will clear it for all of you. Okay. So I saw one of the comments saying that, "Sir, can you teach me C3 cycle, C2 cycle, C4 cycle in one class? In one class, teach me C2, C3 and C4. That is coming up. Okay. So students, only if you like the video, only if you subscribe to the channel, only if you let me know in the comment section how was the class, then only I'll make it reality. Otherwise, not happening. Okay. So with that being said, students, thank you so much. And all of you are aware, question practice is the most important thing. Again, if you go check in the description, there is a all India test series in the description. I'm telling you, every single class, join a all India test series as soon as possible before it's too late that you do not have a time to question practice. So join a test series right now and solve some questions for you. Okay, so thank you so much, students, for today's class. I enjoyed it. Let me know in the comment section how was today's class. Do the homework question in the comments, and I will see you in the next class. Bye bye, take care, and until then, keep practicing, keep studying NCERT, keep solving questions, and you'll get good marks. Okay, so bye bye, take care, students. Take care, all of you.